Good evening, students. I welcome you on the new year 2021. And my best wishes to all of you, to your parents, to your friends, to your near and dears for this new year. And I hope this will bring us, this year will be the best year of our life, students. We can make year 2021 as the best year of our life by making certain good resolutions. The best resolution for us is, students, that we will be more determined, we will work hard, and we will not waste even single moment of life. It's very important. Every moment of life is important, students. It be used for something good. It be not wasted. You can use it for any purpose, doesn't matter. You can use it for your studies, you can use it for any skill, student, music, dance, painting, or whatever your passions are, students. If you are an athlete, if you are a player, you use that moment for the best of you. And I believe if you think, believe, and act, anything is possible. Nothing is impossible, students. You can score full marks in all the subjects. It is also possible. And it is possible for any student. Don't think that one person can do it, others can. No. If one can do it, others can also do it. What is the difference? Hard work and determination, students. So let us begin our new year with the hope that we are going to have more energy, more enthusiasm, more inspiration, and we are going to work more students in this year, and we will have more determination to focus and concentrate on our purpose of life, whatever is the purpose. For the student, the purpose is to, first purpose of you is priority wise to score full marks in the coming papers. Is it right or wrong? Tell me.
Yes, sir.
All right, then my best wishes to you, student. And very important topic, we were talking, student, what we were discussing about the biomolecules, student, biomolecules, those molecules which are making the living system, student, a living system. You are saying that the living system is made up of non-living molecules. What are those? Bio means some big molecules are there, students. And in these molecules, you already talked of carbohydrates, student.
Yes, sir.
What are carbohydrates, students? What are carbohydrates? Can you give me a short answer of that, student? Immediately, what do you mean by carbohydrates? What is your reply, students? We are just warming up the session. Warming up the session. Every game is played. Players warm up, student. You have seen soccer matches, student being played in England. How much warming is going on? Then cricket match is going on. They are warming up the players before they start the final. So we are warming you up, student. You are aware of
Yes, sir.
carbohydrate, student, and you know they are polyhydroxyaldehydes or ketone,
Or
student.
ketone compounds.
And the
Yes, sir.
most abundant organic compound comes on to be the glucose, which you are taking a lot of glucose in fruits and in other stuff which you are eating. And what you find, student, monosaccharides and polysaccharides. The polysaccharides. Can you name one or two polysaccharides, students? Starch, glycogen. These were polysaccharide, students. They were made up of which monosaccharide? Glucose units. You had to more than 200 to 3000 glucose units present in them, students. And can you tell me what is the important linkage which was present in the polysaccharide, students? Very important question. What is the linkage which is present in these polysaccharides in the biomolecules I'm talking about? What is the linkage, student? What linkage is present, student, in polysaccharides? What is your answer, student?
Sir, um, glycosidic linkages.
Glycosidic linkage is a very correct answer given by you. I thank you very much for helping me because that is what I was trying to say. That question is a favorite question in the paper, student. Let me tell you, our sessions are live. Our sessions contain a lot of important and interesting questions which are repeatedly asked. And it helps on in understanding. My teaching of chemistry is not chemistry. I teach you science, students. Science. Chemistry is science. Physics is science, students. That means, students, we have to understand. And there are certain basic things which make up everything, student. If you build a house, you see there are building blocks, students. bricks and all those cement which make this also. When we will talk of biomolecules, they are also made up of certain monomer units, certain small units. So glycosidic linkage is very important. And you know how it was formed. How it was formed. Students, the OH group of one
Yes, sir.
monosaccharide, OH group of another, water out, and there is a oxide linkage, which we call as a glycosidic linkage, student, which is present in starch, which is present in other polysaccharide, right or wrong?
Right, sir.
Cellulose and glycogen are also examples of polysaccharide, students. Then I talked with protein student. Proteins, what are protein, students? They are biomolecules which you are consuming, enzymes, all you are saying they are proteins. Can you give me what is the definition of protein, students? Small things, any question can be there in paper. What are protein, students? Proteins are, what is your answer? You have 30 seconds. My timer is
Yes, sir. Answering.
every time you are getting 1 minute.
So proteins are
Hmm?
are biopolymers made of amino acids.
Amino acids. Very good answer. And what is the linkage? What bond is present in these amino acids? Important question.
So,
What
so
linkage?
bond present.
Which one?
So thinking.
What bond? What linkage is present in these amino acids which make up the protein, student?
Sir, peptide bonds.
Yes, that is absolutely right answer given by you, Kunal. I thank you very much that you are able to recollect. This question has been asked many times. Peptide bond, students. So there are bond, students, which are in chemistry. What is there is a bonding. Some type of bonds are being formed when small units will combine. When monomers, small amino acids will combine, students, to form big molecules, proteins. Therefore, students, the building blocks which we are talking, the biomolecules, students, they are big molecules and protein molecular mass may be more than 10,000 U, which we told you for one mass of one molecule may be more than 10,000 U. So it's a polymer, student. They are all polymers. Okay, polymers. And they are made up of very small monomer units. You have seen that. And there is important linkages which are repeatedly done, glycosidic linkage, peptide bonds, and hydrogen bonds, and sulfide. So many types of bondings take place, which make them very important compounds in our everyday life. And
Yes, sir.
today's lecture is also very, very vital lecture, because here you are, have you heard about student heredity? I am not a bio student, but you are a bio
Yes,
student.
sir. Yes, sir. Yes,
More
sir. In
than
bio.
me. What is heredity, students? What is heredity? You are
So
aware of
traits you. Yes, sir.
what is that?
So basically, traits you acquire from your parents.
Transmission of what? Transmission of certain basic characteristics.
Yes, sir. Traits.
Transmission of inherent characteristics, students. You are having some character, student, inherent in you, where they are coming from, students. They are coming from your parent cells. They are coming from your parent cells, which you are saying
Yes.
heredity. Transmission of inherent characteristics. Inherent characteristics, students. And they are saying that your face resembles your mom, your face resembles your father. Students sometimes they, oh, very, very familiar face, and your characteristics, your properties are also having similar to them in many ways. So that is what you say, there is a heredity, student. Now where the heredity comes from, students, you know that you have in the cells, students, you heard, you have read more than me biochemistry, you have bio, I don't have bio. In the cells, student, there is a nucleus. So the nucleus is there inside the cell. The particles of the nucleus are responsible for heredity. The particles which are present in the nucleus are called chromosomes. Is it right?
Yes, sir.
If I could be wrong, you will tell me more about bio, because I am not a bio student. Chromosomes, students, which are made up of proteins and another biomolecules. These chromosomes contains proteins, students, and also they are made up of biomolecules. So bio means they will be big molecules, students. What are those biomolecules? These biomolecules are nothing but a nucleic acid, students.
Yes, sir.
Introducing a new term, nucleic acids. Proteins are polymers of amino acid, you told me. And they contain, student, a peptide bond. How many? Large number of amino acids are there in the proteins. Enzymes also, you are saying they are a type of protein, students, in the body. Now we are talking of biomolecules, which are nucleic acid. Nucleic acid, when I write the term nucleic acid, that means, student, it is also a polymer. It is a biomolecule. These are also biomolecule, students, nucleic acids. So, in what we find, student, nucleic acids are nothing. When I say what are nucleic acids, students, you are saying they are
Molecules.
biopolymers. Polymers I am using
Polymer.
again.
Yes, sir.
Biopolymers, students. When I use polymer, means they are big molecules. They will be made up of certain monomers, certain small units, and these units will combine. How many? Large number, large number. As you said in, why did I give example of protein? Why did I give you example of starch, student? What we are talking, they are all biomolecules. They are polymers, students. They are big molecules. So certain small units are present in them. They are linked. And the linkage, what type of linkage you have seen? We have already told you. Glycosidic linkage, peptide bonds, hydrogen bonds. All these types of bonds could be there. So students, one thing is clear. First thing, the heredity or the inherent characteristics which are being transmitted from one generation to another generation, there is a chromosomes inside the nucleus, and these chromosomes are made up of proteins
Yes, sir.
and biomolecules. The biomolecules are biopolymers, which we call as nucleic acids. Therefore, student, when I talk of nucleic acid, I will be talking of the two things mainly. One is my DNA, student, DNA. DNA is a nucleic acid. DNA is a nucleic acid, student, and other you are aware of RNA. ठीक है? Right or wrong,
Yes, sir.
student? So,
Right, sir.
what is the full name of DNA, student? DNA stands for, what is a DNA?
Deoxyribonucleic acid.
Deoxynucleic
Oxyribose nucleic acid.
acid.
Ribose. Yes, sir.
Deoxyribose nucleic acid. This is a deoxy. D I have put. And nucleic acid. Na means nucleic acid. Na. This is my Na. Okay. Nucleic acid. Na. Okay. What I am trying to do, just to teach you the basic word by word, what is the meaning of that. We are trying to decode, students, the NCRT and other books. That is our purpose. So nucleic acid, this is also nucleic acid, but it has a D. It has a D. So student, oh, it has a D means what is the D stand for? What you told me? Deoxy.
Yes, sir.
Deoxy. It is deoxy, student. Uh, Deoxy, and they are saying deoxyribo. We will see what is the meaning of ribo, student. These are sugars actually. Ribose, you have heard about ribose is a sugar, student. So deoxyribonucleic acid. Ribonucleic acid. Okay.
Yes, sir.
Trying to understand. And when you said RNA, student, then what is RNA? So RNA is derived from, R is standing for ribo.
Nucleic acid.
Ribo is a ribose. Ribose is a five-membered sugar, student. Five-membered sugar. Glucose is a six-membered sugar, right? Here we
Yes, sir.
have five-membered sugars. Five. So this is a ribonucleic acid. ठीक है जी?
Yes, sir.
Now the first question to you in the exam, that what is the difference between, student, which sugar? These are the sugars, student. These are the sugar. This is the, if the sugar is derived from ribose, see what I am saying. Ribo means what, student? ठीक है? When you are saying, student, if the sugar is derived from ribose, ribose is a sugar, student. What is it? Sugar.
Sugar, sir.
Five-membered. Five-membered sugar. So if it is derived from the ribose, then you, what is, in which it is, which nucleic acid is this ribose present? RNA or DNA?
Sir, in DNA.
I'm saying ribose,
Oh, yes, sorry, RNA.
ribose RIB, so it will be RNA.
Yes, sir.
The first question, student, what is the difference between RNA and DNA? The first question is that the sugar which is present is RNA is important question. Now you start writing notes, student, in terms of question
Okay,
answer.
sir.
So the sugar which is present in RNA is what? Which one? Ribose. And to give it more clear, this is actually beta D ribose. What is this? Beta D ribose,
Yes, sir.
which is the sugar which is present in RNA. That is the name ribonucleic acid. Ribonucleic acid. Okay?
Yes, sir.
And students, if the sugar is derived from, now I'm writing another name, deoxyribose. This is a sugar.
Yes, sir.
This is also five-membered sugar, student.
Hmm.
5 carbons. It also contains 5 carbon, 5 membered, 5 carbon. Okay?
Yes, sir.
So deoxyribose, then in which nucleic acid it is present, RNA or DNA fast?
Yes, sir. Sir, DNA, obviously.
DNA. Okay. Now this is a way to remember it. And this is a question, student. Deoxyribo sugar is a present in DNA. And if I give the complete of this is again beta D. This is called beta D, student. Beta D to deoxyribose. Beta D to deoxyribose. It is a sugar. Okay? That
Yes, sir.
means, students, the sugar is also present in the nucleic acid.
Hmm.
Nucleic acid is a biopolymer. I'm
Yes,
only
sir.
talking one unit, one small
Hmm.
unit. It will be linked to form. Large number of these will be linked. And you know
Yes,
what
sir.
type of bonding we have told you. Somewhere glycosidic linkage, some peptide bond, anything is possible, student.
Hmm.
Bonds.
Yes, sir.
Sometimes they will coil through hydrogen bonds or sulfide bonds, anything. That is a miracle interest of chemistry, student. The bonding is the most important part of chemistry.
Hmm.
The molecules can bond with each other. Atoms can bond with each other through certain, you see, interactions, certain attractive forces, and that is what makes the chemistry most beautiful subject. And you generate new compounds, new substances, students, with the new properties. So, student, a very important question has been discussed. What question I discussed? Tell me. Question is, which sugar is present in DNA and which sugar is present in RNA, student? What is your answer? In DNA,
Uh,
which sugar is there?
yes, sir. Sir, DNA deoxyribose and in RNA ribose.
RNA. Okay, here I have shown you the structure and of the two sugar, students. Two sugars.
Hmm.
Which sugar is this? Beta D ribose. This you can see
Okay.
five membered. This is a Haworth structure. You have drawn the Haworth structure for glucose.
Yes, sir.
Here Haworth five-membered ring. Five-membered ring. How many carbons? One, two, three, four, five. Okay?
Hmm. Yes,
And
sir.
you can see there is an oxygen here. What oxygen is what type of linkage, student, in any sugar? You
Hmm.
know what type of linkage is this? So this is a cyclic structure. Cyclic structure. You know the cyclic structure of glucose also? How it was
Yes,
formed?
sir.
When the in the glucose CHO group combined with the OH group, student, hemiacetal formation. So this is a
Hmm.
cyclic structure of Haworth structure of beta D ribose.
Okay, sir.
The beta D ribose is present in RNA or DNA. Beta D is present where? In RNA or DNA is a sugar which is present in RNA.
RNA. Yes, sir.
And this is beta D 2. Why it is called 2? Deoxy means what? What is the meaning of deoxy? Deox, मतलब oxygen out. Oxygen out. अरे deoxy means what? Now you understand? D means loss of oxygen. At which
Yes, sir.
point?
Yes,
At
sir.
number 2 carbon. At carbon number 2. Got it?
Yes, sir.
So here you can see at carbon number 2, there is a OH group, HOH group present where in ribose. In beta D ribose student at carbon number 2, you have oxygen OH bond. You have this OH, but in here at carbon number 2, there is no oxygen. Is there any oxygen here? Tell me. At carbon 2, this is my carbon 2. So in beta D2 deoxyribose students, the oxygen is missing.
Yes, sir.
That is why we get deoxy. They are the same structure, only the difference is at carbon number 2. Carbon number 2, you have marked it? Important point,
Yes, sir.
student. What is important, I will keep on telling you. So at carbon number 2, this is my carbon number 2 student, carbon number 2. What do you find? The oxygen is absent. That is why I said deoxy. It becomes ribose to D. You want to go from a beta D ribose to deoxy ribose. But at which carbon? Carbon number 2. So I put it 2. Deoxy ribose. Got it? I am converting
Yes, sir.
student beta D ribose into 2 deoxy ribose. Same. Everything is same. Only oxygen is not present here, which was here oxygen is not present. Here you have OH group. Here you have hydrogen. Got the difference? You have hydrogen.
Yes, sir.
You have OH. Try to mark it very carefully. Therefore, students, interesting thing that one difference we know. One difference we know. Now the question is, students, we are saying the nucleic acid is overall name of DNA. They are composed of nucleotides. They are made up of nucleotides. Now I'm using another term, nucleotides.
Yes, sir.
Nucleotide, which are monomers. Nucleotides are monomers of 3 components, students. How many? 3 components. Nucleic acid is a biopolymer. And what is the
Yes,
monomer
sir.
units in them? You are saying that monomer unit is known as nucleotides. And they are made up of 3 components. One is a 5 carbon sugar. You have seen. This is a 5 carbon sugar.
Yes, sir.
A phosphate group. Second, there is a phosphate group, student. And third, a nitrogenous base. So it is a polymer of monomer, which we call as a nucleotide. And the nucleotide contains how many basic components? 3. Which are the 3 components, student, present in nucleotides? Question for the paper. Which are the 3 basic components of nucleotides? Now answer the question. Which are the 3 basic components of nucleotide, students? 5 carbon sugar, phosphate group, and a nitrogen base.
Okay, sir.
If I will have 3 of these together, how they will be linked, we will see that. You know how the things are linking. That is why I discussed of certain type of bonds. We have experienced already in polysaccharides and in proteins. Students, there are 3 basic components of the nucleotides. Nucleotide is a monomer of nucleic acid. How many nucleic acid?
Yes,
I
sir.
think DNA and RNA. The only difference here, we have till now told you the sugar. So there will be a 5-membered sugar, there will be a phosphate group, and there will be a nitrogenous base.
Okay, sir.
These 3 will combine to form a nucleotide. And these nucleotides will then link together. How they will link, we will see, to form nucleic acids.
Okay.
Is it clear up to now?
Yes, sir.
Very important, I am saying. How do we know that? How do we know that they are made up of these 3 components? What reaction we did to know that this polysaccharide is made up of 2 sugars? What reaction was done to say that sucrose contains glucose and fructose? Which reaction you did? Tell me. How do you know sucrose contains glucose and fructose? Tell me. How do you know? Which reaction was done, student? What reaction was studied? I hope you are aware of hydrolysis.
Yes, sir.
Here also we will do the complete hydrolysis, students. What we will do? Complete hydrolysis. So when
Yes,
they
sir.
experimentally chemical composition of nucleic acid, how it is determined by complete hydrolysis of DNA or RNA.
Hmm.
They took DNA, complete hydrolysis. RNA, complete hydrolysis. What they obtained on hydrolysis? Pentrase sugar, phosphoric acid, and nitrogen containing heterocyclic compound, which are called.
Okay, sir.
Therefore, students, when these nucleic acids were hydrolyzed, how many things you got? Three things. What
Hmm.
are those? Pentose means five-membered sugar, phosphoric acid,
Hmm.
a base which is hydrocyclic base, student, which
Okay, sir.
contains nitrogen. That base contains nitrogen. You note it down.
I'm copying the 2 minutes.
Copy everything. You see, one line I write is very important, very powerful. How many questions I have put till now? So many. Seven or
Hmm.
eight questions have been framed from this few lines.
Yes, sir.
And in DNA molecule, the sugar moiety is beta D2 deoxyribose. So the question is, which is the sugar moiety in DNA? Beta D2 deoxyribose. Whereas in RNA, the sugar moiety is beta D ribose. This is important question. You have noted down, students. Noted?
Yes, sir.
Okay. Can I proceed further, students?
Yes, sir. Yes, sir.
Okay.
You can.
All right. Next page. Next page, students. There are two important terms which have been used in the book. One is nucleoside. Nucleoside, student. The nucleoside is a subunit of nucleic acid. What is it? Subunit.
Hmm.
A unit as subunit means smaller unit of a unit.
Yes, sir.
It is a subunit of nucleic acid, you are saying. What are nucleic acid? RNA and DNA. Okay? We know about it now.
Hmm.
This is a hereditary controlling component of living cells. Heredity controlling, student, ठीक है,
Yes, sir.
component of living cell inside your body, ठीक है.
Hmm.
Everything slowly and slowly note down because we don't want to hurry up. Understand, there will be question on this. What is made up of, what the nucleoside is made up of, student? Sugar, ठीक है,
Yes, sir.
plus nitrogen containing organic compound. Only two things.
Hmm.
What is that nitrogen containing organic compound? That is the base we told you, base. Which base? Heterocyclic containing nitrogen. Okay? So
Hmm.
nucleoside has only two things. Sugar and nitrogen containing organic compound. For example, you can see this is a sugar, five-membered. What if
Yes, sir.
this is my five-membered sugar, student? ठीक है, five-membered sugar, sugar containing five carbons,
Hmm.
five carbon sugar, ठीक है,
Yes, sir.
and base. This is the base I have shown. This is linked with the base.
Hmm.
Sugar and base, sugar and base. This base,
Okay.
which base? We have told you heterocyclic, which contains nitrogen. These two things make a nucleoside. What is this? Nucleoside. And what is the, this is a subunit. This is a subunit of nucleic acid. This is
Yes, sir.
hereditary controlling component. ठीक है?
Hmm.
A unit formed by attachment of base. Where the base is present, you can see one dash position. Position of the base is one dash. This is my sugar, student. I have given 1, 2 dash because the base also has carbons. So we, instead of 1, 2, 3, we write 1 dash, 2 dash, 3 dash, 4 dash, and 5 dash carbons. So at the 1 dash carbon is linked student with the sugar mighty base. Okay? So
Yes, sir.
are the nucleosides. In nucleosides, the sugar carbons are numbered as 1, 2, 3 to distinguish these from bases. Clear?
Hmm. Yes,
Now,
sir.
what is a question in the paper? Question in the paper, student, what is a nucleoside? This has been asked in the paper, I am telling you. What is a nucleoside, student? What is your answer? It is consisting of a sugar linked to a nitrogen containing organic compound.
Hmm.
5-membered sugar, and the linkage is at 1 dash position of the sugar with the base. Only base and sugar. There is no phosphoric acid present here. No phosphate. Okay?
Yes, sir.
Then comes the nucleotides. Second, nucleotides, students.
Hmm.
You said that DNA, RNA are often long chain polymers made up of monomers. These are the monomers of the polymers of and DNA, bio polymers. These are the basic units or monomers of student DNA and RNA units. So DNA, RNA, they are made up of monomers known as nucleotides.
Hmm.
These are the monomers. They will be linked in large numbers together to form your nucleic acids. Okay?
Yes, sir.
So when these nucleotides, they are monomers. When they will combine, student, they will be linked together. Okay? The resulting chain, the chain you will get will be polynucleotide.
Hmm.
Poly means large number of nucleotides which are monomers, they are linked together. Now, what is the nucleotide made up of? Each nucleotide is made up of 3 parts.
Yes, sir.
How many parts are there in nucleotides? 3 components, students. In a nucleoside S, this is S, student. How many components are there? 2 components. How many I told you? 2 components. Which one? Base and sugar?
Yes, sir.
Sugar plus base. And nucleotide has 3 components, students. What is that? Nitrogen containing ring structure. Nitrogen containing ring structure, student. What is that? That is called a base. Nitrogen containing ring structure, which I call as a base, student. It is a base, nitrogenous base. Then 5 carbon sugar,
Hmm.
and the third component is, student, at least 1 phosphate group. Okay? Third component,
Yes, sir.
phosphate group, student. Phosphate group is present. And question was asked, student, what is the sequence of these 3? Here was a question, what is the sequence of these 3? 1, 2, and 3.
Hmm.
This sugar is present, student, in the central position. This sugar is present with what position? Central position, student. Note it down. This one. Present in the
Central position.
central position. Nitrogen, phosphate group, and carbon sugar inside that. In between. Just like a sandwich, student. In the sandwich, inside the sandwich, what you put, student? Butter and jam.
Yes, sir. Yes, sir.
Butter and jam. Jam, if I put jam is sugar. How put a layer of sugar inside the 2 pieces of bread, it is inside. So this carbon sugar is present, student, inside the sandwich of base and phosphate. So it is a sandwich of base and phosphate, the sugar. The sugar is molecule, student. The sugar 5. So like you are having 3 components in a nucleotide and only 2 components in nucleoside.
Yes, sir.
Question in the paper: what is the difference between nucleoside and a nucleotide, student? Can you answer that question? What is the difference between nucleoside and nucleotide in the paper? Nucleoside made up of 2 units, nucleotide made up of 3 units.
Yeah, okay, sir.
Only the phosphate linkage is not present. That is base and sugar. This contains base, sugar, and phosphate. What is the sequence? Where is the sugar? Sugar is inside the 2. Which 2 students? On one side you have phosphate linkage, on the other side which group you have? The base. Sugar is in the sandwich. In the
Yes, sir.
sandwich. This is a way to remember, student. So at least one base particle sugar has a central position, student. This question has been asked in the paper. The student didn't know which position the sugar is. Which with base attached to one of its carbon and phosphate group attached to another. ठीक
Yes,
है?
sir.
Up to now, no difficulty. You have noted down. I will proceed further. Follow?
Copy 2 minutes.
Huh?
Copy 2 minutes. 2 minutes.
Okay, note down. There is so many questions still I told you in this. Each thing is a question, student, in the paper.
Done, sir.
Okay, thank you very much, student. Let us see, student, the structure of nucleotide. Now, nucleotide, you are saying nucleotides are the monomers, and when these monomers will be linked to form chains, they will form nucleic acids. That's what we said. Polynucleotides are nucleic acids.
Yes,
And
sir.
how many components you are saying? You can see now. This is component 1. It has phosphate. See, phosphate linkage. This is our sugar moiety, 5-membered sugar, and this is base. Can see 3 things.
Yes, sir.
Base is your phosphate, students. They are linked, and this sugar is the in the sandwich. Sugar is in the,
Sandwich.
because the question was given. What is the sequence? Students will forget. Now you will not forget. In the sandwich, you put sugar, student. Sweet, honey. So, nucleotides are joined together by phosphodiester linkage. What is the linkage? This is called phosphodiester linkage between 5' and 3' carbons of the pentose sugar, student. Pentose sugar. This is a 5' carbon of the pentose sugar and 3'. There will be lower because it is a chain, na? What is it? It is
Yes, sir.
polymer chain. The formation of dinucleotide is shown here. We will show you what the dinucleotide will be there. Therefore, student, this is the definition of a nucleotide. It has 3 components. 3
Yes, sir.
components, student, which you are saying phosphodiester linkage, sugar, and base. Whereas nucleoside consists simply of nucleobase, nitrogenous base, and 5 carbon, ribose or deoxyribose. Because in RNA and DNA, the only difference is the 5-membered sugar is there. One is ribose, other is 2-deoxyribose, means where at position 2 oxygen is not there.
Okay, sir.
Whereas nucleotide is composed of a nucleobase, 5 carbon, and one or more phosphate groups. So the question which was there, student, what is the difference between student nucleoside and nucleotide? This question is answered here. This is the answer of that.
Yes, sir.
Okay? It contains a base and sugar. This contains base and sugar. It contains diester phosphate, phosphodiester linkage, and base and sugar. Three things.
Mm-hmm.
There is this is called a nucleobase, 5 carbon sugar, and one or more phosphate groups. Three things. That only two things. Nucleosides are responsible for encoding, transmitting, and expressing genetic information in all the living cell students. Nucleotides are the building blocks of nucleic and nucleic acids. These nucleotides are building blocks
Yes, sir.
of the nucleic acids. Nucleic acids. Which one? RNA and DNA. ठीक है?
Yes, sir.
And we have told you what is the difference between the two. Only 2 units, sugar and base, 3 units. Will have one more phosphate group also. Here,
Hmm.
I'm proceeding further than this is clear. If you have to note down anything, you can note it down, students.
Yes, sir.
Noted, or you want to note down?
No, sir. Noted.
Noted.
Yes, sir.
Okay, students. Now, students, DNA contains 4 bases because they are polynucleotides. Which bases are present, students? Because question will be there in the paper.
Yes,
We
sir.
are saying DNA contains 4 bases. RNA also contains 4 bases.
Hmm.
DNA, how many bases are there? 4 bases.
Okay, sir.
RNA also has 4 bases. And students, there is a list of the bases. We don't need to know the structures because this is not, we are not studying in so much detail. But the names of these are important, and where the question has been asked, I will be very sure to tell you. The
Yes,
four,
sir.
the 4 bases we have said is adenine, guanine, cytosine, and thymine. They are represented by one letter word, students. A, G, C, T. The 4 bases, DNA contains 4 bases. Which one? What are the 4 bases present in DNA, students? A, G, C, T. What is A? Adenine. What is G? Guanine. What is C? Cytosine. And T is thymine.
Yes, sir.
RNA also contains 4 bases. But there is one difference. That question has been asked. First 3 bases are same. What are the first 3 bases? A, G, C.
Yes, sir.
But the last one, thymine is not there. Instead of thymine, it has U. U is uracil. So, what is the difference now? You can see. This base is not present in RNA, student. Which one? T. So, thymine is present in DNA, but
Hmm.
in RNA, the first 3 are same. But what you have? Uracil.
Yes, sir.
Now, this is a question for the paper, student. This has got a base thymine. This has got uracil. First 3,
Okay, sir.
both contain 4 bases, but one base is different. Which one? Thymine in DNA and uracil
Okay, sir.
in RNA. Question will be like this.
Yes, sir.
Write the name of the base which is different in RNA and DNA. So you have to say thymine is after the 4 bases.
Okay, sir.
Thymine is not there in RNA, instead it has uracil. So here we have shown the structures of these bases. See, they are heterocyclic and have how many nitrogens? See, 1, 2, 3. So many nitrogens are there. These belong to class of compounds. They are called purines in the organic chemistry. These
Yes,
are
sir.
called pyrimidines, students, in the organic chemistry. So that is not in our course. We are not going into full details of this. But any student interested can search for purines and pyrimidines. These three
Yes, sir.
bases belongs to family of pyrimidine, students. and these two belongs to the family of purines. See here you have this 6-membered and 5-membered ring also. Adenine and guanine, they are purines, and these are pyrimidine students. Therefore, student, I am coming to one interesting thing, that nucleotides will mean there is a sugar base and this is my phosphate linkage, diester phosphate. Okay? There are three
Yes,
units.
sir.
Any nucleotide means three things. Which one? Base, sugar, and this. These are the
Hmm.
monomers. They will form long chains, student, to form the two.
Yes, sir.
There is a base. The four bases are present. 5-membered sugar is present. In
Hmm.
RNA and DNA, you know what is the difference of the sugar.
Okay,
Ribose,
sir.
beta D ribose and beta D2 deoxyribose. And one base is different in the two. Which one? DNA has thymine and RNA has uracil and three bases.
Yes, sir.
I can proceed further, students.
Yes, sir. Yes, sir. Yes, sir.
Okay, student, here we have shown the formation of a dinucleotide. देखो, this is one monomer, this is another monomer. Nucleotide, they are linking now. And this linkage will form long chain, students. The linkage linking to form student nucleic acids. Where is the linkage? You can see that. This picture we have taken from the book. You can see this is my sugar, student, and this is a linkage, student. 5 dash at 5 dash position, the sugar, and this is a base. Then at the
Yes, sir.
3 dash OH group, which we told you of, this is linked with the phosphate of the another unit of the monomer, another unit of the monomer, and such linkage will continue. And ultimately, this linkage will lead to a biopolymer, student.
Yes,
It
sir.
will be our nucleic acid. What will be that? Nucleic acid. So in general, in short, student, we are not going into full details of those. That's a part of the biochemistry complete, which is not in our course. So you can say base, sugar, phosphate, then phosphate, sugar, second group, then sugar base, this sugar base phosphate, this type of will continue n times. You see, n times means this chain will continue, students, and form long chains, what we call as nucleic acids, students.
Yes, sir.
So you can see that what are nucleic acids? They are biopolymers of nucleotides, students. We have told you the difference between nucleoside and nucleotide, which bases are different, which sugar is different. This is an important question for the paper, students. All right. Now here is again the story which we showed you, how the linkage is taking place. See, this is a 5-membered, this is a
Yes, sir.
5-membered sugar, student, and this is linked with a base. This is a phosphate group. Then this is another nucleotide. The two nucleotides are forming dinucleotide, and this process
Hmm.
continue. There nobody is asking to draw the structures of these are very big ones, but just to show you, I have taken it. Okay. So students, till now what
Yes,
I
sir.
told you, what I have tried to show you, students, the formation of the nucleic acids. What I have shown, how the nucleic acids are formed, students. Therefore, students, this is the sequence in the chain of the nucleic acids, and the nucleic acids have secondary structure which was given by James Watson Crick. He gave a double strand helix structure for DNA. DNA, student, has a double helix structure. ठीक है? Which
Yes, sir.
in the book, student. It has a double helix, students, in which nuclear chains are wound about each other. These nucleic chains, student, nucleic chains are, student, wound about each other, about each other.
Yes, sir.
ठीक है? About each other, held together by hydrogen bonds. They are held together by hydrogen bonds, students. Important point. How they are held? By
hydrogen bonds.
hydrogen bonds between pairs of bases.
Yes, sir.
Between pairs of bases, student. We have already told you what bases are present, student. The two strands are complementary to each other. There are two strands. They are forming a double helix structure, student, in DNA through hydrogen bonds between pair of bases. And the two strands, these are called strands. Two strands are, students, complementary to each other.
Okay, sir.
Complementary to each other,
Each other.
students. ठीक है? Because the hydrogen bonds are formed between specific pair of bases. Important point.
Hmm.
Hydrogen bonds are formed where? Hydrogen bonds are formed between specific pair of bases, student. Pair of specific pair of bases. Adenine forms hydrogen bonds with thymine.
Yes, sir.
Adenine forms hydrogen bonds with thymine. What I'm saying? Adenine. Now this is important point, student.
Yes, sir.
Adenine will form hydrogen bond, student. With which base? With which base, student? With thymine.
Yes, sir.
Important point to note for papers. Such questions can be there, student. And whereas cytosine,
Mm-hmm.
cytosine is another base, student. It forms hydrogen bond, forms hydrogen bonds, students, with guanine.
Yes, sir.
So there are specific bases with which they are, student, forming a double helix structure. So in secondary structure of RNA, helices are present which are only single stranded. Sometimes they fold back on themselves to form double helix structure. RNA molecules are of three types and they perform
Mm.
different functions. RNA molecules. RNA molecules, students, are of three types. Three types of RNA molecules are there.
Yes, sir.
And therefore, each type will perform a different function, student. So, three types, they perform different functions, students. And what are the functions which they are performing? One, you know, m-RNA.
Mm.
What is that? Messenger.
Yes, sir.
This is your messenger RNA, student. And another one is ribosomal RNA. What they
Mm.
call r-RNA, which means ribosomal RNA, students.
Yes, sir.
And transfer RNA, students, t-RNA. t-RNA, which is called transfer RNA.
Okay,
So
sir.
the DNA has got, student, double strand helix structure. Double strand helix structure, which was given by James Watson, you know, for which Nobel Prize was awarded. Double helix, double strand helix structure, student. This complete diagram is available in the book. Those students who want to learn more about these students
Mm.
and RNA in the NCRT book, they can see, or any other book they will see. So DNA fingerprinting is important, student.
Yes, sir.
What is DNA fingerprinting, student? Every individual has unique fingerprints. These occur at the tips of the fingers, have been used for identification, but they can be altered by surgery, student. A sequence of bases on DNA is also unique for a person. Sequence of bases. sequence of bases student.
Hmm.
ठीक है? Sequence of bases is unique for every person student. Every person. ठीक है?
Yes, sir.
And this is unique, and information regarding this is called DNA fingerprinting.
Mm-hmm.
You know that nowadays they say, okay, bring your fingers with the ink, but those fingerprints can be changed by surgery. But this DNA fingerprinting cannot be changed, student, and these have been used now, where it is being used in forensic laboratories for identification of certain criminal student, identify dead bodies by comparing the DNA of parents or children,
Yes,
and
sir.
identify racial groups to rewrite biological evolutions. So DNA is a chemical basis of hereditary and reserved genetic information. DNA is responsible maintaining identity of different species of organism over millions of years. So DNA is capable of self-duplication during reproduction,
Hmm.
and identical DNA strains are transferred to the daughter cells. So important function of nucleic acid is the protein synthesis. Actually, proteins are synthesized by various RNA molecules in the cell, but the message for synthesis of protein molecule is present in DNA.
Okay, sir.
So there is some biology involved in this, but
Hmm.
from chemistry point of view, I believe for our course, this ends the chapter of biochemistry students.
Yes, sir.
This is all
Sir, now what's left?
we need for the biochemistry student. Now we have what we are having students, inorganic part, inorganic chemistry we have to do with you,
Hmm.
the transition elements, the inner transition element, p block elements. Organic chemistry you have done, and there is
Yes, sir.
a solid state also in your course students. So we will,
No, but sir, it won't come in exam in the pre-board.
I don't know, but all the students have called me and told me that solid state is coming.
No,
So
no. Sir, it's in boards, like it's not in pre-boards.
not in the pre-board.
No, no.
Okay, your pre-board, what is coming in the pre-board students? Tell me.
Sir, I'll tell you 2 minutes. I send you the syllabus. 2 minutes.
Okay, you can send me in WhatsApp
No,
students.
no, no. Sir, I'll so 2 minutes. 2 minutes. I'll tell you right now. Uh, yes, sir. Sir, first 5 chapters of physical chemistry, except chapter number 1 and haloalkanes, alcohol, and biomolecules.
And
And biomolecules.
which part, or inorganic chemistry, what part?
Sir, chapter number 2 to 5.
Huh?
Chapter 2 to 5.
Chapter 2 to 5.
Yes, sir.
What is the chapter 2 to 5 students? Which chapter is that?
So I have to check 2 minutes.
The chapters which we are going to do students in the coming classes. Uh,
Yes, sir. Sir, chapter number 2 is some solutions, electrochemistry, chemical kinetics, and surface chemistry.
okay. And
Okay, sir.
inorganic chemistry, nothing?
And no, and sir, in inorganic solutions, electrochemistry, chemical kinetics, some surface
That is
chemistry.
physical chemistry. That is physical chemistry.
Yes, sir. Yes, sir. Sir, and in organic, haloalkanes, alcohols, and biomolecules.
Okay.
Yes, sir. That's it.
No, nothing of inorganic.
No, no, sir.
Okay.
Yes, sir.
And then what paper is that? Because this is the carrying a large number of amounts in the final paper, in the board paper.
Hmm.
So anyway, what we will do, students, those we will be discussing important questions now from those chapters.
Okay, sir.
Important questions which can be asked in the paper from solutions, electrochemistry, and all those. And after that, we will do the inorganic part, which is left for the final board papers. And in the final board paper, solid state is also coming. Okay. Yeah, in school is not given.
Yes, sir.
No problem. Okay, then students, we will continue with the important questions from different topics which are coming in the papers in the next classes. I thank you. Good night. We will meet you in the next class. Okay.
Okay, thank you, sir.
Okay.